here he is.
Hello?
Hi.
Hi, how are you? Hi. Hi.
I didn't miss anything last week.
You didn't? Oh, yeah.
No.
We didn't have a meeting.
I was I was on Lake of the Woods.
Oh, okay. Yeah.
Just explaining that that's a lake that's between Canada and the U.S. It sort of splits the lake in the middle.
Oh, yeah.
Yeah. So we probably sailed into the U.S. a little bit.
Yeah.
Bradley, can I tell my update if you don't mind?
Yeah, go ahead.
These many days I was having so many bugs related to the implementation of DevoNet actually. So finally I cleared out all the bugs and model was able to train, and the loss function is also less than the last DevoNet models. Last DevoNet models loss was about mean loss was around 1.2 around that, but now loss functions are around 0.99 and it is furtherly decreasing. It is at epoch 0 only. I haven't tried it because of some GPU issues. Maybe by tomorrow I will try it. The code is working fine, and I have completely worked on, and I pushed the code actually. This all code I worked on this week is complete. I just pushed this code a few minutes back, and right now the good thing is SAM model is working and DevNet model is also working. I just need to I think right. I have learned so many lessons while implementing this all code. Writing code is not important, but in a structured way is more important than I think so. So I have implemented everything here, but they are not in structured way. So I was thinking to make it structured to create two different repos for DevOnet model and SAM also, because SAM is mainly used for cell segmenting. DevOnet will be used for acquiring the cell volume, cell area, etc., and cell positions. So why not keep it two different repos for? If we mix two projects in one repo, it is quite confusing actually. I thought combining it into one, but yeah, it's confusing. So I was thinking to bifurcate both of them into two different repos as two different projects.
Okay.
And I do the OpenWorm Foundation thing. And the update would be pretty simple. I have worked on the technical issues this week completely, the loss functions and implementing the SAM model. Have never approached SAM model of training till training, but the model was able to train and learn now, and the loss function is also less as we expected, and maybe the accuracy will be also good too. I just need to add some augmentation to the data. That's it, and make a clear documentation about these two projects separately in a clear way, not in a messy way. Right now, we don't have any pictures, or we just have the code and all these things. So I want to make two different repos for both of them and add some pictures and do a clear documentation. That would be useful for any further projects in future.
Yeah, that would be great. Now you said something about the code being like harder than you think, or what were you referring to there?
Actually, the code, I have done the code part very quickly. I mean, I could solve all the bugs with help of Mayuk and Minuk also. But the situation is there is a mistake from my side. I haven't structured the code in a perfect way. And this is the thing creating problems now. The code was not in a structured way in a two different repos. If you see Bradley, this repo contains of two different models. One is DevoNet, one is cell segmenter SAM.
Okay.
If we keep these two together in one only, it is creating so many problems like dependency issues, etc. So I was thinking to create two different repos, DevoNet separate and cell SAM separate, segment anything model separate, and create a clear documentation to both of them.
Yeah.
I mean, if you see here, the model is working actually. I thought segment anything model will not work at the end, but yeah, here it is working. I mean, this is not the error. I just created a keyboard interrupt. That is the reason it stopped. But it was right now the model was able to learn and it is trying to get the loss function as we expected. Then past DevoLearn models. So my next couple of weeks would be the work of organizing this all code in a correct way and make a clear documentation. And Bradley, I'm really sorry that I haven't attended last week meeting. I was affected with dengue and
Oh,
and
wow.
the hospital has some strict rules that we don't need to use a laptop over there.
Right. Oh, that's fine. Yeah. Yeah.
So I kept away 1 week completely on this thing. But yeah, now the code is working. Everything is working. We need to just make a clear documentation and a paper draft to the whole of this thing. And I have found that Springer has a free publication kind of thing. So I was thinking to write in Springer in applied intelligence.
Okay.
It's mainly for learning and stuff. So yeah, documentation. I have started the documentation. I haven't worked on the documentation yet, but I will start from this week and make a clear documentation. Next couple of weeks would be writing a clear paper about like how the model is working and comparison between the model and etc. Right now, the model was able to learn and it is training also, so there won't be any problem.
All right. Give me a little bit more detail about the data augmentation. What were you thinking there?
Data augmentation, mainly I was thinking, like right now we have around 4,957 images in our data set. So I was thinking to flip the data and in different angles and pad it in different way and applying different filters like it's a grayscale image, right? Why don't we convert into it RGB image? And actually, PyTorch library has different data augmentation techniques. I was thinking to apply those only to this documentation. Data augmentation. Let me show you. I just told these examples of flipping it, rotating in a different angle and feeding it. This would increase the model accuracy and uh, it may create different test cases for the model to learn. Not on a single test case right away image. If the image is flipped in a different way, and if it is fudged, maybe model was not able to learn it. So I was trying to do the data augmentation, apply different filters to it, like a green filter, yellow filter, and just a edge detection. If we apply these filters, it will work definitely fine. I think so. Right now we have the best. I mean, best loss because it is less than one and model was able to learn. If you see here, there is a difference from 0 here. It is decreasing. But if you see here 255, but here you can see that it is decreasing from that. So loss is decreasing and model here it indicates that model was able to learn the whole features from the data set we propose. If we add more data augmentation to it, etc., even the loss function may go below 0.5. That was my idea.
Okay. Yeah, that's great.
Yeah. Next couple of weeks, Bradley, I will completely work on the paper and the documentation part because every project might insisted me to make the documentation a correct way. We are having a meeting, so Saturday and Sunday we are discussing about it. Yeah, he told to make a documentation clearly, and he even warned me that he will fail me if I don't do the documentation in the correct way because documentation is a proper thing we need to learn. He told, and
Yeah.
code also. Actually, I was updating him everything. So yeah,
Okay.
this is the complete work I have done this week, adding the loss functions to DevoNet and trying the cell segmenter, SAM model, removing all bugs. So both of the models were able to learn. So next couple of weeks would be writing a paper and comparing it over different models like UNet, actually past DevoLearn models. That would be my total implementation for a couple of weeks, making a clear documentation and writing a clear paper.
Yeah, I think that's a good plan. I went over some resources on writing documentation. If you're familiar with the meetings, and I think you went to one, that we run on open source on Fridays. And in the Orthogonal Lab Slack, I have a channel. I think you're also in that channel, Google Summer of Code. And I put some resources in that channel. So those are good resources for talking about how to structure documentation. And I know Himanshu's in that channel too. So there's a lot of stuff on how to structure documentation, how to write clearly, of course, but also what to include, and how to make it accessible to people. So basically what we want to do is have like a README file, of course, in the repo, but then also we want to have, maybe some more detailed documentation for, you want to have something for the general public. If you just stop at the repo and you're interested in it, you want to give people enough information to say, this is something I might want to fork, or, star, or do something like that. Then later on, you get a little bit more deeply into the software and you say, well, how does this work, or how does that work? And so that's where the more in-depth documentation comes into play. And then you can link that to like publications. So we've talked about the DevoWorm white paper or the preprint. That can be like, more detailed structure where we have, maybe some examples, some use cases, and then that links to the documentation and saying, see this for more. So it's basically just, you want to make a very accessible version. You want to make a more in-depth version. Then you want to have your publications linked to that, where you really give a lot of detail. But you also you can't give like you can't get into like code examples and things like that in a paper, because it bogs down the paper, So you want to kind of push that into the deep documentation as well. So
I will comment out the whole code because someone wants to see the code. If they don't understand, it would create a problem. So I will add the documentation to the code also, commenting every line. That would be the better thing, I think so. Because right now DevoLearn doesn't have like this of time pop commenting code thing. So I'll try to comment every line what I wrote, and maybe that would be useful when someone reads the code. Go through the code, what is happening in and out. Yeah, that's are my updates.
Okay.
Yeah.
All right. Yeah. And commenting is always important because it helps people walk through it without having to flip back to the documentation. So.
Okay, Bradley. Those are my updates. That's it. Yeah.
Okay. Yeah. Thank you very much for
And
that.
my deadline is also extended. It's still October 24th. I didn't mention in this due to this health issue,
Right.
but it got extended in the main official page. Thank you for that.
Okay, you're welcome. Yeah. All right. Thanks, Sushmanth, for the update. Himanshu, are you there?
Yeah, hello.
Hello.
Can you hear me?
Hi. Yeah.
Okay. I'd like to start my presentation with the things I've figured out for implementing TGNN. So basically, compared to other systems, our topology is dynamic. So basically, our cell diagrams, like the cell nucleus segmentation images and membrane images. So we can find out the topology of cell dependence of time video frames. So we can start from extracting frames and then segmenting, which is the first of our project, which is similar for both me and Sushant. And then we can take a point cloud of that and then do TDA on it. So it will be a static topology if done this. But the hard part is to make it dynamic because the time related image and it's because it's video frames and it's moving all the time. So in this case, our approach would be similar to TGNN. So the first step would be to do the TGNN's implementation and to figure out these two questions, like how GNN can be applied to cell development analysis and how it can perform with TDA on multi-cellular system. And another approach I had thought of was to do persistent homology over point cloud data, which is what my example is built upon today. But it's an image example because I wanted to start with an image and then further go to point clouds. Before that, I will like to explain the topological layer that was presented in the 2022 paper of TGNN. So basically we have a set of, this is a graph, like it's basically a set of node attributes. And here, okay, so this is basically a graph. It's this would be basically the daughter and mother cells in case of like our C elegans data. And this is basically a set of nodes which is in the RD space. Like each topology has like a zero-dimensional space, one-dimensional space, two-dimensional space, etc. And the first step we have to do is to create this node attributes and convert it into a node map, which basically maps each node to some value, and it's basically like a subset of the entire thing. And after doing that, we get like k different views for graph G, because we are applying k node values to a network map. So we are getting like k different views of the same graph. After doing this, this is the main part of the paper is like doing a filtration process, and this is where like a filtration process and on the each and every view, which gives us a persistent diagram. And persistent diagram is like the way persistent homology is like built upon, like the filtration is doing the TG, like the filtration is doing the topological graph computations, and then we are getting a persistent diagram. And the aggregation step is basically to like incorporate it into a neural net GNN layer. So in the aggregation step, it's how we like since like graphs are permutation invariant, so, and it is also permutation. It is a permutation invariant. We have to do this aggregation step, and after we do that, we get an output, and which is basically like the topological graph layer. And since this is a topological graph layer, since it's a layer, we can apply it on any GNN of a type theoretically. So we can also build upon this, which was done in the previous GSOC here, which was basically temporal graph neural nets, which Jiang had worked on. So if we can code this layer and we just incorporate it, so it should work like basically in a vanilla GNN as well. So this is what I had figured out, that this is one of the approaches we can do. And for the example, I have proposed like a function which I thought would be great as an example to show that a TDA works on cell data as well, which is basically doing a low star image filtration. So what a low star image filtration is, that it allows us to express local minimums and maximums if we flip the image, like if you get the negative of the image. And what this does is it gives us birth times as well as saddle points for a zero-dimensional persistent diagram. So the births and these points are basically a good way to stabilize the image and give us the critical points which we can look upon. And this is basically a example like which like of a Gaussian blob and in 3 different spaces, and doing the same thing on this test image will give us a zero-dimensional persistence diagram, where the x-axis shows the birth cells and this is as the death cells. And this at 0.5 we have like a threshold layer, which tells us like it's where the critical point is acting at. So we can convert this entire thing in a cell biology image example, and when we convert this, we have to use a grayscale image, but we already have a grayscale image in our data sets. I've I've worked on both membrane cell membrane data as well as cell nucleus data to see how it changes accordingly. And in this case, what we wanted to give is like a local max of persistence with each cell. And just let's see how it is. So basically, I printed the shape and the values of the original image, and here this is the plot of the segmented image of nucleus data. And after that, I've done the same for our membrane image, and I've done this low star image function on original image, like the memory image, and this is the graph we are seeing here. So as you can see, the lifetime of the cell, it's it's always around nearer to the zero values, and it peaks at a 0.8 value. And this is basically the since our grayscale image is from 0 to 1, like spaces from 0 to 1. This is how this is what the diagram shows us, and as you can see, most of the values are nearer to zero, and it's like it starts to go up after like around 0.5. Sorry, not 0.5. It's 0.05. So that could be like a threshold, which is what I thought of. But to get a good values, what we can do is to smooth the image so that our cells that are neighbors to each other will have like similar values in that range. After doing that, we get like a better. That is what I thought of. It will give us a better output. So after doing that, we get like a more points which are nearer to the zero value, and the lifetime of the cell is also changed. Like first, the maximum was 0.8, and most were nearer to 0.05, and like this. But now it is a little different. So this is just saying that most of the points which we have, we don't need them to give us the persistent diagram that we actually, the topological features that we actually need from it, like the maximum low star filtration points. So after doing this, I've put a threshold as 0.05, and also I've plotted the points which were like above the threshold, and this is the points which we are getting. As you can see, it's nearer to all the points which are like connecting the cells, and I'm getting a little different output. It should be like nearer to the centers of the image, like which is what I'm working on right now. So this is one of the ways we can take a. So this is one of the ways we can basically get topological features from a data set of our choice. Also, one of the things I'm working on, which would be like good for the project, at least like in terms of visualization, is ParaView. So this is one of the tools I've used like during my college for a project in parallel computing. So this is like and this also has a topological toolkit, which is why I was really interested in it. So I was also figuring out like, and this also gives us a lot of topological functions and features. So we can also do topological data analysis on this, and in corresponding to doing TGNNs on it, and we can then also compare both of them, like how we are getting like if we the topological graph layer, how it's giving us persistence data, and also we can get persistence data from this toolkit. And I'm still learning the toolkit, and right now the data which we have for which we use for temporal graphs, it's not fitting inside the ParaView, so I'm figuring that out as well, and I'll get back to you with that as well. Okay. Also, this is my entire update, and my further things would be to look at the TOGL code, which is basically the open source repository which was given by the guys who wrote the topological graph neural nets paper, and from this I'll have to basically do similar Jiang did for his GSOC, that is like to take this repository as like inspiration, and have to create my own TopoGNN library kind of thing, so that I can do it. I can just take I have to refactor this code, and as well as figure out a way to incorporate it in a cell-based data set. So this is like the part which I was mostly confused on, like how will I implement it. So this is And this is what I've thought of, like in the start, which I've in the start where I showed the embeddings and this part.
Okay.
Yeah, so this is my update. So it was a little all over the place because I've been working on it for a few weeks and I was not able to give a proper update. So I apologize for that. But yeah, this is what I've been working till now.
Yeah, that looks actually pretty good. Thank you for the update. Yeah, you've made a lot of progress on things, and it looks
Yeah,
like you've worked out kind of a pipeline for producing
yeah,
something from the data, so that's good. Um, so you're basically plotting the
this,
graph. Yeah, you're plotting the point cloud, and then you're plotting it out, and then you're transforming that into basically topological features, which you could build a graph from if you wanted to. And you're putting that over. You overlaid that onto an image, or I thought I saw that in the last entry in your notebook.
yeah. So I've done the example on the image, and there is another example I'm working on, which is to do it on a time series data.
Okay.
But since our since our data set of the C elegans, which had like the relation between daughter to mother cells, it was also time series based, but it was not entirely like time series based because it did not have like specific timestamps on it. So I had to figure out a way to convert it into a way so that it gives us like time series plots. And once I do that, I can also do a similar filtration method on that graph as well as I can import that into the topological toolkit which I showed earlier. That would be to then.
Yeah. Yeah, I think the time series component would be important to, make something where you could just plug in data and actually have the time series data, and then build some topological features from that. Because we've done a lot of things with static images, but the time series is important for capturing some of these changes.
Yeah, exactly. And also, what we can do is like the Jiang had also given like a website where you can see the entire C elegans tree branch, like from the start to like all the mother daughter cells. I can also try to get that and the data set into ParaView, and I can try to do TD on it. I'll work on that as well this week. I'll just see if this works as well. But
Yeah,
since we have got a pipeline created, I can start working on that. That was like the hardest part to figure out. So I'm happy I could do that.
oh good, yeah. Yeah, the interactive C. elegans lineage tree is what he's talking about. So that's
Yeah.
yeah. So that actually has all the cells, and you can it has like annotation data too. So it tells you what the cells are, and then you can use that as your baseline. So that's good. I don't know exactly how it exports, but yeah.
I have to figure that out. I'll do. Yeah. Thank you.
You're welcome. Yeah, thank you for the update, Manchu. It was great. So yeah, that's great. And then again, the same things that I talked about with Sushmanth in terms of documentation also apply to your project. But I guess you're planning on some papers. I saw you had a reference to maybe a target for a paper in your notes.
Yes, I'll work on that as well. But first,
Yeah.
I have to figure out the implementation part of it, and after that, I have like so I'm still in my final year, so I have like I'll do that as well, like together.
Yeah. Yeah, Sushmanth is kind of working on a accelerated track where he wants to put out all these papers. But yeah, I mean the most pressing thing here is like, having good sort of annotation of the code and then having some documentation. So
Yeah, I have a way to like document the entire thing. Like right now, I'm just using OneNote. But since it's like in particular pages, and it's kind of maintained, and use that as reference. And as also I'll be creating like reports for my college internship reports as well, because that's one of the mandatory things I have to do since it started. So I'll also have that for reference so that I can do it like really well.
Okay.
I think also the oral guys also have a report, I think, in colleges as well for internships.
Okay. Yeah.
Yeah, so it's kind of similar that way only. So I'll have to do that, and I can show it to you as well, like what I've posted over there. All right, thank you.
All right. Thank you. Yeah. Great.
Thank you for my time.
Yeah. It looks like Dick and Jesse have joined us since I last got a break in here. So welcome. Yeah. Hello. So yeah, those are our GSOC updates, and I've been working on the paper. Actually, it was Lucas and I were working on the paper for the differentiation codes. So we were actually doing a lot of work on analysis, getting the analyses together. So this is where we're doing all the methods and getting the data together. And so I talked to Lucas Friday, and he's done a lot of work on this so far, and we compared notes. So we're coming along on that, and yeah, I don't know if Lucas is going to join us today, but I think we're pretty close to getting to a point where we can review the document. I don't want to do it today. It's still not quite to the point where I can walk through it, but it's coming along. Some pretty interesting findings about some of the things that we're looking for. We're doing so. To recap, for those who don't know, we're doing this bioinformatics analysis. We're looking for sort of target genes that Dick and Natalie mentioned in their book that might be involved in differentiation and in a putative cell state splitter. So we have the protein list. Lucas has been busy getting protein matches from NCBI. He's been doing blast searches, looking for a list of proteins and their homologs and paralogs across 4 species, actually. So we have yeast, we have Drosophila C. elegans, which is our target, and then mouse. And so that corresponds to, okay, yeah, thank you, Himanshu. So we have C. elegans, which is our sort of focal species. We have Drosophila, which is another example of mosaic development. We have mouse, which is regulative development, and then yeast, which has no development. So there's no developmental period there. And the idea would be those proteins would be, have different counts for those different species. And then in C. elegans, I'm looking at some of the genes that code for those proteins. So if you go through the literature, you can find examples. In C. elegans, it's a wonderful library. They have not only the sequenced draft genome, but they also have a lot of function for different genes. And, so there are papers where people have found genes coding for proteins. Yeah, so great, but please leave time for the rest of us to review what you found. Yeah, so we should have this done by the end of the week, or to a point where it's like a draft document where we can go through it. So let me share my screen. I have a couple things to share with you today. The first I wanted to talk about are a couple references going back to some of the synthetic biology stuff we were talking about, and some of the maybe even early life stuff we were talking about. So the first thing we were talking about was this coding guide to synthetic biology. This is something that Nico McCarty put out. Nico McCarty he does this site on. He writes this thing called Codon. It's a Substack that has a lot of interesting content. But this is a nice guide he's put together on synthetic biology and some of the readings that you might need to know if you want to engage in this area. So I shared this in one of the channels in the Slack, and Morgan, who's not here today, really thought it was a nice reference. And so that's good. This basically just gives you different references to different areas of synthetic biology. So this is going to be a living document. This is something that's going to be updated over time. So let's see. So there are a lot of seminal paper links here. There's the basics of synthetic biology. So Drew Endy's paper on the foundations for engineering biology, which is way back in 2005 now, but that's on the list. A brief history of synthetic biology, which was published about 10 years later. And synthetic biology doesn't just go back to 2005, it goes back to a lot of the work that's been done on building constructs to deliver things like fluorescent proteins or gene inserts and things like that into cells. So a lot of this stuff with CRISPR comes out of a long history of modifying cells, putting genes into cells, knocking in genes, even knocking out genes, things like that. So that talks about that. Then there's synthetic biology applications come of age. So a lot of this is not just for experiments where you show a difference if you take a gene out or put a gene in. These are gene copies, or sometimes they're the gene itself, but you have these applications that you want to build. So, different applications that are useful to agriculture or to medicine. This one is the cell really a machine? This is from the Journal of Theoretical Biology. This is interesting because a lot of synthetic biology is predicated on this notion that the cell is a machine and that we can work with it like a machine. We can take parts out and put parts in and make corresponding changes. So there's always a correspondence between like a parts list and the function. So that's, of course, an assumption that people make, but is it really a fair assumption? And so they talk about that in this paper. Then the rest of these references in this section are, just kind of talking about research tools, which change very frequently. So some of these, papers are maybe a little out of date, even though they're only maybe about 10 years old. There's some forward-looking papers, which is now not I guess it was, it seems like a forward-looking paper, but it's actually a retrospective. The second decade of synthetic biology, 2010 to 2020, which was published in 2020. So it kind of talks about that. And then, the second wave of synthetic biology for modules to systems where, we think of sometimes like genetic systems as modules, like gene regulatory networks, groups of genes that work together to produce some maybe phenotype or function. But, thinking beyond the module. So if there's a module for some, to produce some enzyme, that's great, but that fits into a larger system. So that fits into the system, the genome of the cell and the function of the cell. So all these things are sort of, they have to go from modules to systems, and that's this paper talks about. Then they have this section on the central dogma. We'll talk about this paper. 60 years ago, Francis Crick changed the logic of biology. Some things on the central dogma, some references on gene expression and regulation. So these are really for the uninitiated. But some of these are really kind of interesting, like this unified theory of gene expression, which is kind of a reinterpretation of what people are, people think about like the relationships between gene expression and proteins. And so a lot of the things that we've been discovering, especially with respect to synthetic biology, are they have this sort of, they've outstripped the theory that we have for it. So now people are trying to come up with new theories around this. This paper is actually about 20 years old, so I don't know what the follow-up has been, but we'll talk about this later as well. Then there are biological networks and mathematics. So this is only a few papers in this area, but you have things like artificial gene regulatory networks, mathematical modeling in synthetic biology, modeling and analysis of gene regulatory networks, and so forth. Then there's those graphic notations. So this is kind of getting into systems biology and some of the things that people are doing to represent things graphically. So, some of these things involve like, open standards, There's the synthetic biology open language, which is a community standard for communicating designs. So in synthetic biology, you're say like designing a gene circuit, you're designing how it's supposed to be put together, and you need a unified language to build that. It's kind of like programming, where you have a unified modeling language, but they have a different way of doing this with synthetic biology. Then there are these graphical notations that you can use. There's open language visual, which is an open source graphical notation, and so forth. Then there are technologies. So these technologies are always changing. There are always new technologies coming out all the time. And some of these, though, have stood the test of time. So DNA sequencing generally is, a similar process for the last, 40 to 60 years. So sequencing doesn't necessarily change. It's just the way in which we do it. There are different techniques for doing it. So there's this paper, DNA Sequencing at 40. This is by Jay Shandure. This just kind of talks about some of the different sequencing techniques that have been used in the last 40 years. Next generation DNA sequencing methods, which if you get into next gen methods, you'll find that there's just this massive proliferation of methods. So this reference is from 2008, but I think it sort of puts a lot of the methods into order for people so that they can understand what methods do what. So some methods sequence DNA and do certain things to the amount of gene product, quantify them. There are different ways that you can quantify them. There are different ways that you can sequence the DNA. There are also methods for quantifying and sequencing epigenetic characteristics, things like that. So this is there's kind of a zoo of different methods here. And it may be a little bit out of date because there have been a lot of methods since 2008. In any case, it's probably a good reference to go into. Then there's DNA synthesis. So people in synthetic biology need to synthesize DNA, synthesize new sequences. So there are a number of papers here. DNA assembly, where you're assembling fragments of DNA and putting them together. There are other types of methods of cloning. This is cloning genes, not cloning organisms, but there are different ways you want to clone a gene, DNA cloning, molecular cloning techniques, isolating, cloning, and sequencing DNA. So there are all these ways that you can do this. This is basically PCR and associated methods. There's CrisprCas, which is, of course, this method where you can target different stretches of DNA and either cut them out or put in new pieces. And so this is important in synthetic biology because you're trying to build these circuits. And if you can put something into next to an existing promoter or something, it helps you build a circuit. And so there are different ways you can do this. And this gives you, CRISPR-Cas is a very popular method for sort of modifying genes, building new function into gene circuits and whatnot. So this is a number of different references on this. It's a relatively new method, maybe not relative to a lot of molecular biology, but like in terms of biology, it's recent. It's since 2015 really. There's base editing, which is editing DNA bases. There's prime editing. There's directed evolution, which is where they try to evolve things towards a goal. This is a technique that's used a lot of times in proteomics. Protein structure prediction and design, genome synthesis, and then applications. So there's biomaterials, which is a popular area. There are a number of references here, a number of review articles. Biosecurity and biocontainment, biosensors, which are actually useful for things like arsenic biosensors and other types of cell-based biosensors, sensors for water contaminants, things like that. Then there are these CellFree systems, which I'm not really familiar too much with CellFree systems, but they do have a number of readings here. And then gene circuits. So of course you have to understand gene circuits. It's sort of like the building block of synthetic biology, and they have a number of references on how to build gene circuits. And those all come from the Lac-Operon model, which was developed in E. coli. And so they've been, this is like the basic level of sort of gene regulation and understanding it and manipulating it. And so, of course, this goes beyond the Lac-Operon. You can do a lot of things in different organisms, but in E. coli, yeah.
If someone wants to know about the Lac-Operon, they have from North Dakota State University, they have a virtual cell, and they have a cartoon of the whole Lac-Operon series.
Oh, yeah.
And it's very understandable.
Oh, great. That's great. Thank you. Yeah, so actually in E. coli, you can do a lot of really interesting things. You can engineer E. coli to see light. You can engineer them to do different things, to produce different gene products. And there have been some interesting experiments where people have engineered bacteria more generally to do a lot of cool sort of like lighting up or spelling out things in a dish or whatever. So there are a lot of really interesting like demos you can do and interesting things you can build. So this is not just theoretical. Like people build things all the time now with largely with bacterial genomes because they're easier to work with. But so then there are gene drives, which are this is a paper of genetic control of mosquitoes. So there are ways you can build gene drives that are allow you to control things like different. These are a lot for mosquito mitigation, and so. But there's of course some things you need to be aware of, like some issues here. So there are engineer microbial communities. There's metabolic engineering, plant engineering, recoded genomes, synthetic cells. So synthetic cells, that's another really interesting area. And this is where they're building cells from different types of very minimal cells. And they're able to build sort of the molecular aspects of the cell and plug them in. So this is programmable fusion and differentiation of synthetic minimal cells. This is transmembrane transport in inorganic colloidal cell mimics. So these are things that are just containers for the cell or for the molecular stuff. And the thinking is that if you go back far enough in early life or, to some very simple organism, the cell is basically this container for a lot of these molecular reactions and things like that. So, this is, again, an area that is being innovated upon all the time. So, speaking of that last point, there's an article in Quanta magazine. I think it's a recent article on these type of minimal cells, and the title is "Even Synthetic Life Forms with a Tiny Genome Can Evolve." So, by watching minimal cells regain the fitness they've lost, researchers are testing whether a genome can be too simple to evolve. And so this paper, let me magnify this a bit more. So this paper talks about this system. So 7 years ago, researchers showed that they could strip cells down to the barest fundamentals, creating a life form with the smallest genome that still allowed it to grow and divide in the lab. But in shedding half its genetic load, that minimal cell also lost some of their hardiness and adaptability that natural life evolved over billions of years. That left biologists wondering what the reduction might have been a one-way trip. So this reduction may have been a one-way trip. In pruning the cells down to their bare essentials, have they left cells incapable of evolving because they could not survive a change in even one more gene? So basically what they're saying is that if you have this sort of minimal genome, and this is something they do in a lot of this area of synthetic biology, where they find these minimal genomes, and it's usually a bacterial genome, where they do these combinatorial knockouts. So they basically knock out every gene until they, and the gene either allows the organism to survive or it doesn't. If it doesn't allow it to survive, then it's considered an essential gene. If it does allow it to survive, it's considered sort of a superfluous gene or a redundant gene. And so from that type of experiment, you can assemble a minimal genome. And so this is what they're getting at, this minimal genome. And if you knock a gene out of that minimal genome, it can't really survive a change in a gene because theoretically it's sort of locked into that. It needs everything to survive and there's no path forward. So this is, Dick says, what if vesicle changes its volume to near 10 to the 11 times, or 0, 10 to the 11 times? Consequences, opportunities? Not really sure what that means, but near 0. Okay. So if you change the vesicle volume to near 0, as you're saying. Yeah, I don't know. It's a good question. Early Earth spun fast. So this goes back to the stuff we were talking about with early life. So yeah, if your daytime is very short and your cell is very small, yeah. Well, I don't know. Yeah, it's a good question. I'm not sure. Yeah, 10 to the 11th during the origin of life. Yeah. So in this paper, they talk about some of this work on minimal genomes and these minimal cells. So they say minimal cell, they mean minimal genome. And so now we have proof that even one of the weakest, simplest self-replicating organisms on the planet can adapt. During just 300 days of evolution in the lab, the generational equivalent of 40,000 human years. So this is where they're actually testing this time. So they're actually looking at, like they're doing, when you do laboratory evolution, usually pick organisms that have a very fast replication time. With DNA or even with bacteria, you can do this pretty easily because the replication period is, within maybe a day or so, or maybe less than a day. In 300 days, you can actually simulate or you can experience 40,000 human years if you were to take like human evolution and compare it with the evolution of this genome. 300 days would equal 40,000 years. Measly minimal cells regain all the fitness they've sacrificed. So this is a team from Indiana University. This is a paper in Nature, Evolution of a Minimal Cell. And they talk about this in this paper. The researchers found that the cell responded in selection pressures about as well as the tiny bacteria from which they were derived. A second research group at the University of California, San Diego came to a similar conclusion independently in work that had been accepted for publication. So it turns out that life, even such simple wimpy life as a minimal cell, is much more robust than we thought. So if we have just this minimal set of genes that are essential for the cell to survive, and you start evolving it, you put it down the path towards, replicating and mutating and things like that, eventually you'll end up with a more robust genome than you started with. And so you can start off from a point of absolutely no redundancy, just the essentials, and you can end up with this variety that allows you to have genes that are sort of redundant and other things as well. So you can actually have this very rich genome that aren't all, not all the genes are essential. You can throw rocks at it and it's still going to survive. Even in a genome where every single gene serves a purpose and change would be seemingly detrimental, evolution molds organisms adaptively. So that's basically the article. This is the minimal cell from the Craig Venter Institute. This is SYN 3.0. So this is a strain of bacteria developed in 2016. They work with this a lot. This is a minimal version of the parasitic bacteria Mycoplasma mycoides. So they strip down the genome of that organism, and they have this cell line now that they use. And so this is this is kind of what we're talking about. So this is a bacterial genome. This isn't metazoan genome. So metazoan genomes or any kind of linear genome is much different in a lot of ways. So, but I don't know of any experiments people have run on those. Mostly these have been run on bacterial genomes. Going back to the papers I promised you before, this is the first one. 60 years ago, Francis Crick changed the logic of biology. So this is a nice historical piece on Francis Crick's lecture in September 1957, in which he outlined key ideas about gene function, and in particular what he called the central dogma. And these ideas still frame how we understand life. This essay explores the concepts he developed in the influential lecture, including his prediction that we would study evolution by comparing sequences. So this is sort of the historical base for a lot of the work we talked about in the synthetic biology reference page, talking about these different things. And really a lot of this work on molecular biology comes down to what was proposed in this lecture. So this originally entitled Protein Synthesis. The title acquired a magisterial introduction during writing up for publication the following year. The lecture went far further than the title suggested. As Crick pointed out in the opening paragraph, he also addressed the other central problems of molecular biology, those of gene action and nucleic acid synthesis. So this is kind of what was put together in what was later called the central dogma, or the central dogma lecture, for it was here that he first publicly presented this frequently misunderstood concept. So basically it's the idea that things go from DNAs to transcripts to translation to proteins. So information moves out from the DNA to the proteins. And so he he sort of outlined this because before the 1950s, we didn't really know that DNA existed in the double helix structure that we came to know. And so you also needed a theory to put together a lot of the, DNA and protein and some of the other things in the cell. So this is where this it wasn't really a theory, but it was a framework for thinking about this. It also saw Crick outline his view of the nature of life and of genetic information and the source of protein folding, as well as making two bold and spectacularly accurate predictions. The first was that there must exist a small adapter molecule, now known as tRNA, that could bring amino acids to the site of protein synthesis, and then in the future scientists would be able to explore the rich evolutionary sources of information by comparing sequence data. So in the first point, he basically predicted tRNA, which exists in the ribosome. So you get transcribed DNA. It goes to the ribosome. It gets translated or it gets transcribed by tRNA into amino acids that then get built into proteins. And a lot of this happens at the ribosome. And there's some fascinating work that's been done on these tRNA pools in the ribosome and how they can be selected for different codons or how they're biased towards certain types of codons. And this happens in cells in all different species. Sometimes it's species specific, sometimes it's cell specific bias. But this is, this is sort of the basis of all this work. The second part is that we should be able to explore evolutionary origins by comparing sequence data or, evolutionary sources of information. And so this is the other part that he talked about. So, yeah, he talks a lot about the central dogma. He sets up this framework. He builds a model of, and the central dogma is a lot more complicated and nuanced than I just talked about. So, but they basically outlined all of this. So he outlined the sequence hypothesis and protein synthesis. So they kind of came up with this idea that DNA is the structure in a double helix, but they also found that there are these chemical bases that form a sequence. And so these sequences then are transformed into these proteins, and so they were able to link all that together. In doing so, he took these ideas and found that the data increasingly suggested that RNA was some kind of intermediate between DNA and protein. So we fit RNA into the model. These data refer to ribosomes rather than mRNA, but they figured out the sort of the structure, basic structure of this, and developed a scheme to explain the relations between these three classes of biological molecules. In doing so, he had to get to grips with what exactly was a gene and what took place if DNA was used as a template for RNA. So this is, again, putting all these things into the proper place, and this has stood the test of time. So this is not just a biochemical problem. This is an abstract thinking problem. This is a model that you have to build. So that's a nice article. This other paper is the Unified Theory of Gene Expression. So, as I said, a lot of these findings in biology are sort of just findings, but you have to put a theoretical net around them. Now, with synthetic biology, you wouldn't think you would need to do that. You think you just need better tools. But in fact, you also need better theories. And so in this case, they propose a unified theory of gene expression. This paper is about 20 years old, so it's not a recent paper. I don't know if this has stood the test of time or if people have developed upon this, but let's see. The human genome has been called the blueprint for life. This master plan is realized through the process of gene expression. Recent progress has revealed that many of the steps in the pathway from gene sequence to active protein are connected, suggesting a unified theory of gene expression. So this is, taking a lot of the things in the cell that are going on and kind of casting a theoretical net around them. So this is an image of things in the cell. This is the traditional view of gene expression. So gene expression occurs when you get a transmembrane receptor. Something binds to that receptor. There's a signal that's sent downward into the nucleus through the cytoplasm. This is transcription factor activation. It goes through the nuclear pore into the nucleus. It triggers chromatin, which opens up a gene. Then there's this promoter region, which gets initiated. Then there's this elongation and termination, these steps where the gene is transcribed, and then a mRNA product is produced. And so then this is where you get this 5' 3' orientation that mimics what you see in the gene and how the DNA is laid out. Then you get this capping, you get this processing of the RNA, so you get this secondary structure, mRNA packaging, and then it's exported from the nucleus and sent out into the cytoplasm. Then there's translation, which occurs at the ribosome, which we just talked about, where the mRNA is translated into amino acids through these tRNAs, sort of taking the templates and stamping them out into amino acids, and then matching what's on the mRNA template. And then you get proteins that emerge from those amino acids, and then protein folding. And then proteins are active, and that's where you get. So this is the standard sort of model based from the data and what we know. Now, a unified theory of gene expression would kind of bring this together. So they talk about some of these steps. This is kind of like a process model. And then, kind of arriving at a theory, you would come down to, I think it's not 2, but 3. So this kind of goes through a lot of this process. And, you kind of consider some of these steps as different, with different insights. So ordered DNA binding and chromatin remodeling at promoters, insights into a chicken and egg scenario. So this kind of talks about how the requirement for chromatin decompaction for transcription factor binding to DNA appears to create a chicken and egg scenario. These factors induce local chromatin remodeling, but their interaction with DNA requires prior chromatin decompaction. So how then do transcription factors access the DNA in the first place? And so you can consider this, you can do this experimentally, but you can also do this theoretically and think about different examples and how they play out. I'm not really sure this qualifies as sort of a unified theory. I'm never really comfortable when people throw that around because it's just kind of like bringing all the pieces together. Whether they actually came up with a sound theory is another question. But this kind of, at this point in history, this was when there was a lot of excitement about molecular biology. So it wasn't quite that we had like, I mean, the human genome had just been sequenced. So, we're entering a period that will revolutionize the study of gene expression. We know enough about the individual steps of the gene expression pathway to begin to understand the connections between them and the processes that regulate them. The traditional view of a cell as a bag of molecules separated into compartments by membranes is beginning to be challenged. I guess that was the whole point of this, framing this as a theoretical question. And so, we know the parts, we know the process, now we need to build the theory. Maybe a theory of a cell is too ambitious. Maybe we need to dial that back to like a theory of the nucleus or something and then move from there. So now I'd like to talk about a few papers related to the molecular biology of differentiation. So this is relevant to our paper on differentiation codes that we're working on, including one paper that answers a question Dick asked last week or 2 weeks ago about asymmetric divisions. So let's get started. The first paper is on E-cadherin, and this paper is titled An Instructive Role for C. elegans E-cadherin in Translating Cell Contact Cues into Cortical Polarity. And so this paper talks about cell contacts providing spatial cues that polarize early embryos and epithelial cells. So basically, when you have early differentiation in the embryo, you need spatial cues. And when we talk about spatial cues, we're talking about things like anterior-posterior polarity. We're talking about polarity during cell divisions, things like that. Now, cell contacts, or the contacts between cells, provide these spatial cues. So, for example, when you have a cell that has polarity, so if you have, let's take the two-cell example from C. elegans. Okay. So these will eventually go down to their respective sublineages. Now, if you notice that these two cells have contacts here, so there's a polarity here that allows it to sort of maintain this linkage. And this is what they're talking about, these cell-cell contacts. The polarity is anterior-posterior, and these are the cell contacts. And so E-cadherin is a protein that allows contact-induced polarity in many cells. So as this two cell starts to form more cells in each part of the anterior and the posterior end, get more cells that sort of emerge from these two sublineages. There are cell-cell contacts here that are important, and E-cadherin is important in maintaining these cell-cell contacts and in maintaining the polarity across the embryo. This is one reason why these embeddings that Himanshu was talking about are important, because we can build these kind of models and look at the things that mediate these connections between cells. However, it is debated whether E-cadherin functions instructively as a spatial cue or permissively by ensuring adequate adhesion so that cells can sense other contact signals. So is it a spatial cue, or is it just a signal of sort of the touching of the membranes of the different cells, adhesion? That's an important point, because if it's a spatial cue, then there should be some long-range function to it as well. And it certainly should be that if maybe you took a cell out, if there was a mutant phenotype, for example, where there was a cell missing, that there would be maybe a gap in the embryo. Or, the cells migrate a lot after they divide, so maybe their migration, if you took out the E-cadherin, maybe their migration would be screwed up. Something like that. That's what they're getting at here. They don't know whether it's a spatial cue or some sort of adhesive function. In C. elegans, contacts polarize early embryonic cells by recruiting row gap PAC1, which is another protein, to the adjacent cortex, including PAR protein asymmetry. So you have asymmetry of how proteins are expressed. Here we show E-cadherin HMR1, which is dispensable for adhesion, functions together with alpha-catenin and HMP1, the P120 catenin JAK1, and previously uncharacterized linker PIC1 and this binds PAC1 and recruits it to the contacts. So you have a series of proteins, a protein cascade that needs to be in place to realize this sort of cell-cell contact, to realize the function of ECadherin in its context. Misallocating the HMR1 intracellular domain to contact-free surfaces. So this is where we have this HMR1 that's mislocalized. And so this is in a contact-free surface, so it's just expressed somewhere else. Draws PAC1 to these sites and depolarizes cells, demonstrating an instructive role for HMR1 in polarization. So the HMR1 is an interesting protein and associated gene. And I'm going to be talking about it more in the paper. I just recognize the name as something that's going to be important for regulating some of the developmental genes. But in any case, we'll come back to that later. Our findings identify an ECadherin-mediated pathway that translates cell contacts into cortical polarity by directly recruiting a symmetry-breaking factor to the adjacent cortex. So this is an interesting kind of paper that talks about some of these pathways. ECadherin mediates this pathway. It involves a lot of proteins being in place. And when they're not in place, we don't have this function for ECadherin. So one of the things we're trying to do in our differentiation code paper is talk about ECadherin as sort of one of these proteins that is necessary or that should show up as a product of different cell differentiation. And indeed, these cell-cell contacts are an important part of cell differentiation, because when these cells divide and they migrate, they need to be in contact with one another, but they also need spatial cues to move them around the cell. What their findings are here is that they sort of have this spatial specificity, but they also have this sort of sensing aspect. So you have this complex cascade of proteins that need to be in place. If you move the protein expression to another place on the cell, it doesn't operate as needed, so it has to be spatially localized. You can see that, like, in cells, they need to have basically this spatial coordination amongst themselves. And it's based on the expression of these proteins, where they're localized on the membrane. So for example, if this cell here started to migrate in this direction towards the midline, then it would come into contact with new cells, and it would need to make contacts with those cells, and that would need to be regulated in a different location than if it's making contact with the cells further anteriorly to the anterior end of the embryo. So migration, of course, this differential expression of all these proteins will play a role in facilitating this migration. So their answer here is a little bit of both from the original question. And so they do some experiments here. These are basic experiments where they're looking at the different things that are involved in this mechanism for ECadherin. They look at moving, expressing it in different places. These are some microscopy images. So this is PAC1 structure function analysis. This is the PAC1 locus here. And they show the pH and gap domains, the position of XN6 nonsense mutation. So they look at it for different mutations. They look at this, the stain here. So this is for PAC1. These stains, you can see along the edge of the membrane, you have this expression for PAC1. This is, of course, important in this ECadherin pathway. And you have PAC1 here in green fluorescence. You can see it as well. And so the results are the PAC1 N-terminal domain mediates cell contact localization. So we have it's cell contacts, but it's localized cell contacts. A homophilic adhesion protein HMR1 contributes to PAC1 localization. P120 catenin JAK1 and alpha catenin HMP1 recruit PAC1 to HMR1. So we have this cascade where things are recruiting each other. And then you have HMR1 GFP localization. You have PAC1 localization. So you can see it's very similar. And then this is sort of a diagram of what's going on at the cell-cell contact. So where the cells join together, this is a cell-cell contact. This is where you have HMR1 ECadherin. So you have two of these proteins in each cell, and they're aligning. The JAK1 P120 catenin, this is next to this ECAD in this cell. HMP2 beta cat is next to the ECadherin in this cell, and then HMP17 alpha catenin is below the beta catenin. So this is all happening within one of the cells. In the other cell, it's also happening, and then the HMR1 ECadherin are the ones that come into contact between the cells. So that maintains the contact between cells. And then within each cell, you have this cascade of other proteins that are sort of joined to it. It's interesting because on our list, we had ECadherin, we had beta catenin, and we had, I can't remember if there was any other. I think those are the two that are really they're kind of joined at the hip anyways, functionally. If you read through the literature, there are other ways in which they're connected. And so this is an interesting sort of context to this. Okay. So that's the ECadherin paper. Now I wanted to talk about another paper where it's a more general paper about cell fate specification in the C. elegans embryo. So this is about cell fate specification as opposed to sort of cell-cell contacts and some of these cell-cell interactions. So this is from Morris Maduro, and the abstract reads, cell specification requires that particular subsets of cells adopt unique expression patterns that ultimately define the fates of their descendants. So this is where cells are specified for by their gene expression patterns, and you can identify those using bioinformatics techniques. Usually it's the upregulation of certain genes, and we kind of think of those as a unique fingerprint or signature of a certain cell type. Now, you could critique that method and say, well, that's only one aspect of it. Maybe there's a lot of overlap with different cell types. And indeed, people compare different profiles. And sometimes those are unique. Sometimes they share genes that are upregulated. But in any case, this is a requirement of cell specification, that some genes are upregulated that maybe have a very specific functionality to a certain cell type. Of course, there are going to be things like housekeeping genes that are upregulated. But these expression patterns sort of prime the cell for the fates of their descendants. So in C. elegans, of course, we know that the lineage tree is deterministic. You go from the top down to the terminally differentiated cells. And all those cells have to be in place. They also sort of are precursors to that terminally differentiated cell. So we know, for example, that part of the lineage tree, for example, will produce germ cells, or a part of it will produce neurons or muscle. And so in those precursor cells and those founder cells and some of their descendants, you'll get an elevated gene expression pattern of these fates early on before the cells actually show evidence of that fate. So that's the kind of thing we're looking for. And this is also true of mammalian cells where you have different precursor types. You get these upregulation signatures in sort of the precursors to a certain cell type. So you have neural precursors, for example, or muscle precursors that aren't exactly the terminal fate, or at least the fate that we recognize them as, but they do express some of the things that will later go into making that identity. And so in C. elegans, cell fate specification involves the combinatorial action of multiple signals that produce activation of a small number of blastomere specification factors. So this is where we go back to the blastomere and we talk about what kinds of things are specified. Factors just meaning the genes that are expressed. So we just pick a number of genes that are upregulated, and these are this is activation of these blastomere specification factors actually sort of facilitate these signals to sort of drive forward the differentiation process. These initiate expression of gene regulatory networks that drive development forward, leading to activation of tissue specification factors. So we have blastomere specification factors and tissue specification factors. And it's interesting because in C. elegans, we think of it as a deterministic lineage tree. So it basically unfolds the same way in every organism, unless you have a mutant, in which case maybe a cell is missing, or it very rarely gets misspecified. But when you do, you don't get a normal phenotype. And so this is an interesting thing because it's saying there are two steps to this. There's blastomere specification, which kind of puts it on the road towards differentiation, or keeps it on the road towards differentiation. It facilitates a lot of signaling between cells and signaling within the cell. And then these tissue specification factors, which are sort of, what tissue does it belong to? What tissue is it going to belong to? It primes it for being in a context where its neighbors are of the same tissue type, or that is part of a larger tissue with other cells participating. In this review, the C. elegans embryo is considered as a model system for the studies of specification. So it's not really like, a mammalian system where we have sort of, a cell will commit to a fate, but it can be driven away from that fate. So if you take a cell that's maybe a muscle cell and you put it into bone, or if you take a bone cell and you put it in a muscle cell, it could convert into a bone cell or a muscle cell. So it can convert into the tissue that it's a part of if you do a transplant experiment where you take a cell of one type, you put it into another type of tissue. Now, it's an interesting point because it doesn't always work that way. So in mammals, you have to have some level of pluripotency in order to get that conversion to be complete. If you get a cell that's really firmly differentiated, it may actually not convert fully, or may undergo apoptosis. So even in the everplastic mammalian models, you see this specification that's hard to break. In C. elegans, it's a nice system because you don't have this sort of plasticity. You do have cell specification once the cells are or even before the cells reach their terminal differentiation. They're in this sort of mode where they're going to be, they're priming to be the cell type. And so, nevertheless, you can see the mechanism. The techniques used to study cell fate in the species and the themes that have emerged are described. So this is from Developmental Dynamics. This is from the year 2010. So I'm not really sure if there's been a lot of innovation in this area since then, but this basically lays out a lot of how to study this. So, they talk about metazone embryonic development. Embryonic cells must ultimately assign fates to individual cells and tissues. When a cell has been specified for a particular fate, it is capable of generating those tissues if cultured away from the remainder of the embryo. So basically, if you take a cell out of context, if you take it out of its embryo, if it's been specified for a particular fate, it can generate tissues. It can generate, say, muscle cells if it's been primed for the muscle fate. So you see this in cell cultures where you have cells, you may expose it to muscle factors, or it might be pretty far down the pathway and you put it in a dish and you can get muscle cells. So, a lot of cell differentiation is priming by its molecular environment, its microenvironment, as well as just kind of this process, this internal process. But if it depends on how far along the way it's primed. So if it's been primed for, say, a muscle fate, it will reach a muscle fate. If it's not interfered with by any other type of fate, so if you put a muscle cell, something that's been primed to be a muscle cell, into a culture of neurons, it probably will convert to some sort of neuron, but maybe not. It really depends on how far down that path of commitment that muscle cell has gone. As all embryonic cells are derived from the zygote by mitosis, cells must acquire differences in gene expression over time. In most animal embryos, this is achieved through a combination of inherited maternal determinants. So there are a number of maternal determinants that are inherited in C. elegans and in mammals and invertebrates. And so you have, in most animal embryos, this is achieved through a combination of inherited maternal determinants, cell-cell interactions, which we've talked about in the context of EK-adherent, but also other interactions such as juxtacrine signaling and paracrine signaling. Positional information, which means that the cell kind of has positional information about where it is in the embryo. So, for example, if it's in a location where muscle should be, or if it's surrounded by muscle-like cells, it will be more likely to become a muscle cell. And factors segregated within a lineage from a mother cell to its descendants. So this is where you're in a lineage in the C. elegans lineage tree. It's a deterministic tree, meaning that if the mother cell is, a certain type, or it comes from a certain founder cell, the daughter cell is restricted to those types. And this is true of a lot of different cell types where you get this fate restriction where, the daughter cells can't just become another type of cell altogether. So, for example, if you're in one of the muscle-making lineages, which you can actually map out on a lineage tree, you're not going to suddenly have the cell become a germ cell. It has to become what its maternal cell and maybe what its founder cell allows for. In modern developmental biology, the description of gene activities that result in specification constitutes a gene regulatory network, or GRN. And so this is GRN, Sensu Davidson and Irwin. So this is the type of GRN where, if you've ever seen a Davidson style GRN, it's basically a circuit diagram of different genes and their products and how they interact. So these are kind of, they're hard to implement computationally, but they're really descriptive of the biology and how these things. I don't know if they have any pictures of them here, but this is the kind of GRN, because there are many types of GRNs that people have proposed. So these are gene regulatory networks. They basically link genes together by one gene regulating another gene, or one gene producing a product that regulates another gene. So this is, although animal embryos have evolved different ways of specifying very early embryonic cells, the properties of GRNs are similar across many systems. Hence, the study of cell specification in model systems can be used to exploit the advantages of that system to reveal new mechanistic insights into general GRN properties. And so this is one of the things we're interested in, are these gene regulatory networks. And we've talked about them in the group in the past. It's been a while since we've talked about them, but I think it's always worth talking about them because they're very useful. They're very central to developmental biology. So C. elegans is good for looking at sort of this generational aspect, looking at embryogenesis, because you have a short generation time of about 3 days. So you can generate a lot of embryos in 3 days. And so you can actually get a nice sample of things that you can look for, things like, lineage trees, and you can look at like developmental gene expression. So originally Salston mapped out the lineage tree, and he was able to demonstrate that C. elegans really doesn't show very much variability from animal to animal. It's what they call nearly invariant. So there's some plasticity in the lineage tree, and it's mostly post-hatch or post-embryonic. So these cell lineages give a really nice model of sort of non-variability. So this is an example of a lineage tree. You see these sublineages where they produce different outputs. So the AB lineage produces epidermis, neurons, pharynx, muscle. MS produces pharynx, muscle. E produces cells for the gut. C produces muscle and epidermis. D produces muscle, and P produces germline. So notice that a lot of these sublineages, these are what they call founder cells. So we don't see the founder cells with an AB, but they're founder cells here. There's MS, E, C, and P. Those founders, there are 8 founder cells, and those founder cells kind of define what the subset of, or restricts what tissues are going to be made from that sublineage. So this is kind of like in, fate restriction in vertebrates, where you have sort of this process of where you go from totipotency to pluripotency to certain types of precursor cells that can produce certain types of cells. Once you're at the precursor cell stage, you need to use some sort of engineering technique to get yourself out of that precursor stage. But even so, there's this sort of restriction of fate that occurs over time in a lot of these systems. So in this case, we have cells from the zygote, which is totipotent, to some of these founder cells, which are pluripotent, but actually the lineages that are going to emerge, the daughter cells that are going to emerge, are only going to be restricted to certain types of cells. And in fact, in the adult, you have this nomenclature of specific cells that come from this lineage tree. So there isn't a lot of plasticity here, but there is this fate restriction property. Notice that their muscle gets produced in a lot of these sublineages, but some lineages are really highly specified. So E, for example, is highly specified for gut cells only, which is the stomach and some of the intestine. D is highly specified for muscle, and so there are different types of muscle that these sublineages produce. MS is basically the pharynx and muscle, so this is like the area where the C. elegans is ingesting things and the muscle surrounding it. P4 is the germline, so that's highly specified. And then within AB, you get more of a mix of things, but it's largely epidermis, neurons, pharynx, and muscle. So all these things are produced. They're restricted in their fates, but they're producing sometimes they're only producing one type of tissue. Sometimes they're producing multiple types of tissue. It's an interesting kind of aside, but it's interesting how this is structured because, some of this, of course, your C. elegans lineage tree is organized from anterior to posterior. So, some of the stuff going on in the posterior end of the worm is maybe more specialized than the anterior end. Now, that, of course, is kind of absurd because the bulk of your connectome is in the anterior portion of the worm. But, those neurons, of course, this neuron category is, it includes a lot of diversity. So I don't want to say that, this is just a bucket of neurons. There are a lot of different types of neurons. And of course, when you get down into the lineage tree, there's specialized types of neurons. And when you get down several levels of sublineage, you get specific types of neurons that emerge. And so that's basically how this works. And then this is an example of kind of this process from the 2-cell, the 4-cell, the 8-cell, to what they call this comma stage, which is where you have this, it looks like a comma, and the gut is forming at this point. And then you get this, so it's a fold in the embryo, and then you finally get the gut inside, you get the pharynx, the muscle on the outside, so you have these outer layers of muscle, which are all these sublineages, and then the germline, which they don't show, which is kind of like right here. And it's just the eggs and the vulva. So this is how this works in C. elegans. So it's a very nice, elegant system for this type of thing. And then, so this is the cell specification gene list. So this kind of talks about these different sublineages and some of the proteins that are associated with them. So, you get for the germline, you get this Pi1, the CCCH zinc finger. This is the P lineage, this is germline, PAL1 caudal, C and D, MED1, 2, MS, and E. So you're just taking things out of the literature and finding connections to these are all cell specification genes. So these are all genes that are active when that cell is being specified into a certain, it's being restricted to a certain fate. So these are some of the proteins involved in blastomere identity, These are some of the proteins involved in tissue identity. These are some of the proteins involved in maternal blastomere identity. So there's a maternal and a zygotic component to blastomere identity. And then you have the tissue identity, which is, of course, this later step. And so here's the analysis of gene expression and perturbations in normal development. So you get this gene expression that occurs normally. You can also perturb the embryo to modify the gene expression to make sure that you understand the mechanism. And so this shows some examples of what they're doing here. And it just vindicates the model that they're working from. So another thing they do is this forward and reverse genetics. So these are complementary approaches. So there are different ways you can do this, different methods. In addition to embryo manipulation, cell-cell interactions can also be studied genetically. C. elegans possesses a hermaphrodite mode of reproduction, which is mostly how it reproduces. There's some sexual reproduction, but it's limited. This has greatly facilitated the recovery of mutations in developmentally important genes. The first regulators to be identified that affected early embryonic fate specification events were recovered in forward mutagenic screens that looked for maternal effects in embryonic lethal phenotypes, exhibiting a deficit of tissues of one type and an excess of another. So you do these genetic screens where you screen for mutations. They call them mutagenesis screens. You look for embryonic lethal mutations, so that means that they produce dead embryos, and then you make the connection. So you can actually do these mutagenesis screens as a form of forward genetics. And then so you can do these kind of screens followed by mapping and molecular cloning. This has been a good way to identify these regulators. Screens like this have recovered GLP-1, which encodes a notch-like receptor important for asymmetries in the AB lineage, APX1, which encodes a delta-like ligand that mediates a GLP-1-dependent cell-cell interaction between P2 and APP, which is, we saw P1 and AB, ABP and P2 are descendants of those two cells. So this is in I think the 4-cell stage where this alters the fate of ABP, and so these interactions are altering the fates of different cells given a mutation. So what they're doing is they're finding all these mutations that affect fate, basically, that are important for setting up the fate restriction. So they've had a number of examples here. Regulators can be identified by their expression in an early blastomere lineage. PAL1, which encodes a caudal-like homeoprotein, was first identified by zygotic mutants, later found to play a central role in specification of sublineages C and D. The somatic descendants of P2 following the discovery that PAL1 protein is found in the early P2 and EMS lineages. The same thing holds true for those. Similarly, TBX35 was identified as a candidate MS factor for the MS lineage because of its expression in the early MS lineage. So all these things, we're trying to link them, we're trying to implicate them in this fate restriction in these sublineages. And then you can use things like RNA interference and knowledge of the genome sequence. So we know we have a draft genome sequence. It was actually completed a little bit before the human genome in 1998. And there's been an update to this genome sequence in 2019, which refined it a bit. But you basically have to have knowledge of your genome, your whole genome, and you have to have a map so that you can do RNA interference, so you can target certain regions of a gene to knock its expression down and remove its expression, and then see if it produces a phenotype that's viable. So forward genetics is where you're actually looking at mutations that have occurred. Reverse genetics is where you're inducing some sort of change to the genome or to the phenotype, and you're looking at the effect. And so, there are a number of methods you can use for these sorts of things. You use transgene reporters, of course, to get a signal that represents the presence or absence of that gene or protein being expressed. And so these are all things that, you can use for this type of thing. I do find this quite a fascinating paper because I think there's a lot of things we can learn from it in terms of looking at this differentiation code. So we'll be coming back to that paper perhaps in the differentiation code paper or later on in another meeting. But I'd like to finish up with this paper, and this is actually something that it was a point Dick raised in one of the previous meetings about asymmetric division events, and of course these are important in looking at the differentiation tree because we're interested in asymmetries and we're interested in differentiation. And so the title of this paper is Asymmetric Division Events Promote Variability in Cell Cycle Duration in Animal Cells and E. coli, which is a bacteria. So asymmetric division events are interesting because they will sometimes produce one sort of dead cell or, one dead cell and one live cell, or sometimes they'll produce cells of different sizes. So there's basically some asymmetry being introduced into that cell division. If it's a cell division that's, where there's one dead cell and one live cell, then that dead cell will be, there won't be a pair, there will only be one sister cell. So that has implications for a number of things in the organism. Also, if you have a difference in size, and sometimes there is a significant difference in size between cells during a division, that can also be, have an effect on the later organism. Because the daughter cells will eventually become mother cells, and they'll either make a contribution to their daughter cells that compounds over time, or if it's a larger size cell, that size differential compounds over time. So it's very important for, say, like an embryonic development. In E. coli, it's a little bit different because basically it's a clonal reproduction. But still, you have this issue of, its effect on the colony or its effect on its descendants or whatever. So asymmetric cell division is a major mechanism generating cell diversity. As cell cycle duration varies among cells in mammalian tissue cell culture cells, we asked whether their division asymmetry contributes to this variability. We identify among sibling cells an outlier using hierarchical clustering on cell cycle durations of granddaughter cells obtained by lineage tracking of single histone 2B labeled MDCKs. Remarkably, divisions involving outlier cells are not uniformly distributed in lineages. So this means that you have these asymmetric cell divisions. They're not uniformly distributed in lineages. They appear to emerge from asymmetric divisions taking place at non-stochastic levels. So these are, I guess they're targeted in different lineages in different places. These asymmetric divisions are sort of not randomly distributed across the lineage tree. A parent cell influences with 95% confidence and 0.5% error the unequal partitioning of the cell cycle duration in its two progenies. So this is where, like I said, these things compound. So the progenies carry on these sort of the effects of this asymmetric division. So when you have an asymmetric division, say one daughter cell dies and the other lives, those daughter cells will produce progeny, of course, that not only live, but carry on, maternal things from the mother cell, maternal factors, or the complement of chromosomes that were segregated into that mother cell all those divisions ago, and they get carried down into the progeny. There's also, there's also information that's lost by that dead cell. The same thing for cell size. If you have a larger cell, the daughter cells are also going to be larger. The progeny are also going to be larger. And what they're kind of saying here is that this is not a random process. This is very specific, and it may have a specific functional reason, but we don't want to go there necessarily. So upon 9 and downregulation, this variability propagation is lost, and outlier frequency and variability in cell cycle durations in lineages is reduced. So there's the cell cycle duration aspect. So the cell cycle duration, you'll see this in the C. elegans lineage tree sort of later on, at least by the 256 cell stage, you start to see these asymmetric lengths of divisions. So up until that point, the divisions are relatively constant, but you start to see variability. And this is part of this priming that we talked about in the last paper, but this is also due to some of these asymmetric divisions. So this is actually talking about downregulation as in the progeny, as the lineage continues. This variability propagation is eventually lost, and outlier frequency and variability is reduced. As external influences are not detectable, we propose that a cell autonomous process, possibly involved in cell specialization, determines cell cycle duration variability. So this is where we're talking about sort of the fate of the cell as it's being primed, as it becomes a terminally differentiated cell. There's the cell autonomous process that takes place. And so that's what they're talking about in this paper. And so, this kind of goes through what we've been talking about. And so, let's see. So one of the questions they ask is, we wondered whether the variability in cell cycle duration of related cells originates from intrinsic processes segregating asymmetrically in divisions. Our working hypothesis. So their working hypothesis was that the variability in cell cycle duration of related cells. So there's going to be this asymmetric division, there's going to be this variability in cell cycle duration. Does this originate from intrinsic processes segregating asymmetrically in divisions? And that's what they're kind of expecting. To test this, we searched for recurring cell cycle motifs in the lineages. First, we computed all possible mother-daughter pairs, sibling cell pairs, which are daughter-daughter pairs, and pairs of cells among granddaughter cells. So, if you look at this in terms of a lineage tree, we have three sets of relationships. We have a mother-daughter relationship, which is where we have a cell, and we have a mother cell, we have two daughter cells. Then we have four granddaughter cells that are like this sort of, this third generation. And then you have pairing. So we have the mother-daughter pairs, which are two. So you have a mother and a daughter here and a mother and a daughter here. You have the daughter-daughter pairs, which are these two daughters. And then the granddaughter pairs. And so I guess you could take these like combinatorially if you wish. But that's what they look like. We investigated whether cell cycle durations between the members of each of these pairs were rather the same or very different from each other. This was also done in Sandler et al., reference 21. The similarity of the cell cycle durations is highest between daughter cell pairs, followed by granddaughter cell pairs, and the least between mother and daughter cells, as reported. So this is like where you have these this variation. So where this effect wears off over time is what they're finding. And so the similarity of cell cycle duration is high between daughter cell pairs followed by granddaughter cell pairs. Actually, that's not what they're saying. What they're saying is that there's a similarity between the granddaughter cells, similarity between the daughter cell pairs, and then the least between mother-daughter cells is because it's an asymmetric division. As cells of the same generation rank are thus more likely to be similar. So if you're in the same generation, you should be more similar. Diversity motifs among these cells are particularly meaningful. Further, as diversity is among granddaughter cell pairs, as this is higher than among daughter cells, we looked at whether these pairs form special motifs. We therefore generated all granddaughter sets, a set of 4 cell cycle durations of the granddaughter cells of the same grandmother cell, and analyzed whether these 4 cells are always very alike or show, relative to each other, reproducible diversity motifs in their cell cycle durations. The theoretically most minimalist diversity motifs among the cell cycle durations of 4 cells are the following 2. So what they've done is they've taken these groups, they've looked at their sort of similarity. They're similar within groups, and between groups they're least similar. So it's kind of a little bit of a confusing point because you're doing this analysis of comparisons. But basically what they're arguing is that in each of these groups you have motifs. You have these diversity motifs. So the first one is a 3-to-1 motif where one of the cells has a very different cell cycle duration. That's the outlier cell relative to the other 3. So if we go back to the granddaughter cells, we see that there are 4 cells. And those 4 cells, there's a similarity that marks maybe 3 of them. And the 4th one actually shows an outlier behavior. So it might take longer to divide. So that's where you're getting this asymmetry. So that's one motif. Then the other motif is a 2-to-2 motif, where the cell cycle durations are more similar within a pair than between the pairs. So this goes back to the granddaughter cells again. You have, these are the pairs, the granddaughters from a specific daughter. And so those 2 would be comparable. And so the 2-to-2 means that 2 of these will be outliers compared to the other 2. So that means that one of the daughters is passing down this timing difference. The timing difference will be the same in both granddaughters, say from daughter 1, and daughter 2 it'll be the same, but between the descendants of daughter 1 and daughter 2 they'll be different. So that's what they mean by diversity motifs and by outliers. The behavior is an outlier if it's different from the norm, and it happens in these different ways. Recall that all the granddaughters are descended from the same mother cell, so they all inherit similar things. It's just that this change happens as sort of an outlier, so that the asymmetry is an outlier behavior. To capture our lineage as such motifs that are based on relative differences within a granddaughter set, we use the outlier detection method. In this case, the 4 cells of the granddaughter set cannot be separated into 2 clusters with a given threshold. Method assigns the granddaughter set a flat motif. So they were interested in this 3-to-1 diversity motif. And this motif is also remarkable as one of the 2 daughter cells that gives rise to the 3-1 granddaughter set divided into an outlier and a non-outlier cell. The 3-1 motif occurs in 2 manifestations. The first is the 3L motif, where one of the cells has a long cell cycle duration. So a long cell cycle duration is basically where the division time is longer. So cell cycle is really setting a lot of the gain for this process. So let's take an example here. We have 2 daughter cells, daughter 1 and daughter 2. And from daughter 1 we have 2 granddaughter cells, daughter 2, and then those cells divide. So daughter 1 divides at a certain rate, and this is maybe different from daughter 2, or maybe the same from the mother. And then the cells in granddaughter 2, their progeny are dividing at a certain rate. We're not really interested in that right now. We're interested in granddaughter 1 and granddaughter 2. And as you can see here, there's a difference in time, the length of time at which these descendants are generated from each granddaughter. Now, in granddaughter 1 and granddaughter 2, this time is dependent on cell cycle length. So there's a whole circuit, there's a whole GRN, as it were, for cell cycle and regulating cell cycle. And so that period is going to increase in granddaughter 1 versus granddaughter 2. That's what they're talking about with this increase in cell cycle. It's just the amount of time it takes for the cell to go through a division. And so obviously, if it goes through a shorter division, it can produce progeny more quickly. So this is where one of the cells has a long cell cycle duration relative to the other 3. And so in this case, your 3-to-1 motif is that all of the cells, even the other granddaughters of D2 here, have the same length of division, short divisions, and then GD1 over here. So this is 3 and 4. GD1 over here is a long division. That's what they're talking about, that motif. And then the 3S motif is where one of the cells has a shorter cell cycle duration, S cells, relative to the other 3. And so this is where it's the opposite of this, where SD or GD1 would have a short cycle and the rest of them have a long cycle. So this is, this is all dependent on cell cycle and modifying that circuit, modifying that GRN. And so this is part of this whole model. Specifically, each granddaughter set in the 36 SI non-target lineages was ordered by magnitude of the cell cycle duration, and then hierarchically clustered based on Mahalanoboi distances. So this is a method where they, evaluate the clustering method. So this is actually, I think, where they're doing this clustering, and they show these different cells. So if I'm not mistaken, this is actually tracked image lifetimes of cells with indicated cell identities originating from a single cell over 75 hours represented in a lineage tree. Nodes depict cell divisions. Edge length reports tracked image lifetimes of bijectively assigned cell identities, which corresponds to cell cycle durations of these cells if the cell could be imaged over the entire lifetime. So basically this is the length of time for the cell cycle. These are the different cells, and you can see this asymmetry emerge over time. You have a symmetry here, and then you have slight asymmetry here, and a longer period of asymmetry here. And so you can see this compounds across these different sublineages where you get these asymmetries introduced. And so this shows that this is a compounding process. And this is kind of a way they have to do this from imaging. But they're able to reconstruct this. So this shows the diversity motifs, this figure. This is actually where they show some of the things I was diagramming. This is the long division. This is the short division. And you notice it's 1 of 4 granddaughter cells. This is the 2 to 2, where you can have it in, from one daughter, or you can have an asymmetrical division in both daughters. So in each case here, daughter 1 produces this is what I was trying to diagram originally, but I didn't understand what they were getting at here. This is D1 and D2, so this is like a short and a long from each daughter. And so this is, again, another way that this can occur. Then there's, of course, the flat, where there's no variation. Then they show some of these experiments where they actually get an empirical measurement in the experimental data, and then they permutate these trees to show some of these motifs. And so this is where they're getting some graphs from some of these experiments. Then they show this dissimilarity index versus frequency. And so you can see the empirical examples here. This is 9N, which they referred to in the abstract. And you can see differences. You can see that there's a lower frequency in the 9N and higher frequency in the non-target. And then, you have this flat expression here, where in the flat case, where there's no asymmetry. And so it's quite a bit different. So this is, they kind of walked through this. This is a nice simulation of asymmetries and things like that. And this is, so now they talk about the non-stochastic distribution of S and L cell divisions. So we asked whether a cell intrinsic processor noise contributes to the generation of the 3-to-1 motif. So is it just the noises like causing one of these to be short, one of these cell division lengths to be shorter than the other 3? Because it seems like that might be a possible thing, that noise could affect it so that maybe you have 1 outlier out of 4 or 1 outlier out of 64. So this is an important question to ask. To this end, we probed whether a cell with an outlying cell cycle duration propagates some effect onto the cell cycle durations of its 2 daughter cells in the next generation, thus affecting the 3-1 motif frequency in the next generation. So to do this, they analyze the positions of the relative outlier cells and the lineages, comparing them with all the other cells, termed zero cells. And the following propagation test reveals whether cell cycle duration of daughter cells are uniformly at random linked to mother cells, which we can define here as a stochastic association, or a non-uniformity exists, which we define as a non-stochastic association between mother and two daughter cells in terms of cell cycle duration. So they basically found that there's this absence of detectable external influences. This is not an extrinsic thing, it's an internal thing, or an intrinsic thing. And this expression of as 9n erases the statistical influence of parent cells. So this is something that they've also show in this figure. So that's all for that paper. Thank you for paying attention. Hope you learned something.